protein synthesis really work? There are two main parts to the process, and the first step is transcription. This is when DNA is transcribed into mRNA in order to leave the nucleus. The first step of transcription is initiation. Pretend that this man, named Dan, is DNA, and let's say that he's a professional candy-making man who owns his own factory.
Every day something jumps in the way, gets complicated. I feel devastated by what I'm feeling.
So here's Dan's house, which represents the nucleus. Right now, there's a snowstorm going on outside, and Dan is trapped inside of his house. But he remembers that he has an important candy deadline to make.
But there's one thing I remember, I only wanna be with you. Life sounds like...
Here we are inside of Dan's house, and here is his wife, Rose. Let's make her RNA polymerase. Dan has to get to the factory in order to reach his deadline, because he has all the information needed to make the candies. So he convinces his wife, Rose, to let him outside, but Rose tells him that he has to wear the right gear in order to leave the house. Out of nowhere, Rose goes to hug Dan, kinda like how the RNA polymerase attaches to a Tata box, which is the promoter region on DNA. Next we have elongation. So here, Rose helps out by dressing him from the bottom up with a snowsuit. Let's let this represent how RNA polymerase unzips the DNA and how it builds a complementary strand in the 5' to 3' direction in DNA. Now Dan is ready to leave his house because he has transcribed or changed into proper gear. Oh wait, but before he steps out of the door, he needs his super snow boots and a super protective snow mask. This is a step known as modification, where the boots added are like the 5' cap, and the mask is a poly A-tail.
Be with you. Life sounds like...
So here is Dan making his way through the storm to get to the factory. While Dan is driving, he erases all of the unnecessary information in his head and only keeps the information relevant to making his special candy. This phase represents introns and exons. Introns are considered to be sections of the pre-mRNA that do not code for amino acids, and exons are sections that do code for amino acids. Introns are cut out of the pre-mRNA by ribonucleoproteins, and exons are spliced together by these proteins. Now we move on to the next part of protein synthesis, translation, which is the actual protein making phase. The first step here is initiation. So let's make the factory act like the small and large ribosomal units that attach to the mRNA. Side note, ribosomes are made up of rRNA. So Dan the Candyman goes into the factory and sees that there are two other workers that are setting up a special robot to make the candies. Now we move on to elongation. Here we have Dan with the robot, which is responsible for carrying the candies, just like tRNA carries the proper amino acid. The robot, which is tRNA, goes into 3 different sites. In the A site, Dan scans his 3 fingers on the robot to read his fingerprints for the instructions. This is like a star codon, which is 3 base pairs. The robot now has the proper candy or amino acid attached to it. The robot then moves to the P site and connects with the other pieces of candy to form one long chain of candies.
So say Geronimo, say
Next, the robot goes to the E-site, where the candy is no longer attached to it and is released from its duties.
Geronimo, say Geronimo.
And now we are at the final step, termination. When Dan the Candyman has reached his desired amounts of candies to be made, he stops scanning his fingers. The man gives a go to the workers to release the candy from the robot. This represents a stop codon, signaling in a release factor protein.
And you rush to me and it sets us.
The long chain of candies is then coated with caramel and brought to the packaging site to get wrapped, like how the release factor proteins bring in a water molecule to hydrolyze the polypeptide and release it from the enzyme. Dan the Candyman is now happy that he has completed his candy deadline. Yay! Candy for all. And that ends protein synthesis, my friends. Hope you enjoyed this video.